Proteins are essential molecules found in all living cells. They are the workhorses of the cell, carrying out a wide variety of functions that are crucial for life. Proteins are involved in almost every aspect of cellular function. They act as enzymes, speeding up chemical reactions. They provide structural support, helping cells maintain their shape. Proteins also play vital roles in transporting molecules, fighting off infections, and transmitting signals within and between cells. Our bodies are incredibly complex machines, and proteins are the intricate parts that keep everything running smoothly. Imagine a bustling city. Proteins are like the builders, architects, delivery drivers, and security guards all rolled into one. The journey of protein synthesis begins with a process called transcription. This is the first step in gene expression, where the instructions encoded in DNA are used to create a functional product, like a protein. Transcription takes place inside the cell nucleus where the DNA is housed. The DNA molecule contains genes, which are segments of DNA that code for specific proteins. Think of DNA as a master blueprint stored safely in the cell's headquarters. During transcription, the DNA double helix unwinds, exposing a single strand of DNA that serves as a template. An enzyme called RNA polymerase binds to this DNA strand, ready to transcribe the genetic information. Imagine a scribe meticulously copying information from a sacred scroll. RNA polymerase acts like the scribe, while the DNA strand is the invaluable scroll. As RNA polymerase moves along the DNA template, it reads the sequence of DNA bases, adenine, guanine, cytosine, and thymine, and adds complementary RNA nucleotides to create a new molecule called messenger RNA, mRNA. mRNA is a single-stranded molecule that carries the genetic information from the DNA in the nucleus to the ribosomes in the cytoplasm, where protein synthesis takes place. Think of mRNA as a messenger carrying a copy of the blueprint from the headquarters to the construction site. Before mRNA can leave the nucleus, it undergoes a few important processing steps. These steps ensure that the mRNA molecule is stable and ready for translation. One crucial step is splicing, where non-coding regions called introns are removed from the mRNA molecule, leaving only the protein coding regions called exons. Imagine editing a film. Splicing ensures that only the important scenes make it to the final cut. Once the mRNA molecule reaches the ribosome, the next stage of protein synthesis, translation, can begin. Translation is the process of converting the genetic information carried by mRNA into a chain of amino acids, which will eventually fold into a functional protein. Ribosomes are like protein synthesis factories, and they require the help of another type of RNA molecule called transfer RNA, tRNA. tRNA molecules act as adapters, bringing the correct amino acid to the ribosome based on the sequence of codons on the mRNA molecule. Each codon is a sequence of 3 nucleotides on the mRNA that codes for a specific amino acid. Imagine tRNA as skilled workers, each carrying a specific building block and knowing exactly where to place it based on the blueprint's instructions. Translation involves 3 main steps: initiation, elongation, and termination. During initiation, the ribosome assembles around the mRNA molecule and the first tRNA carrying the amino acid methionine. Elongation involves the addition of amino acids to the growing polypeptide chain, and termination occurs when the ribosome reaches a stop codon on the mRNA, signaling the end of translation. Section 5. Protein folding, achieving the perfect shape. Once the polypeptide chain is complete, it detaches from the ribosome and begins to fold into a specific 3-dimensional shape. This folding process is crucial for the protein's function. The sequence of amino acids in the polypeptide chain determines how the protein will fold. Think of it like origami, where the folds and creases determine the final shape of the paper crane. Specialized proteins called chaperone proteins assist in protein folding, preventing misfolding and ensuring that the protein reaches its correct conformation. Chaperone proteins act like helpful assistants, guiding the protein to fold correctly and preventing any mishaps in the process. Section 6. Post-translational modifications, fine-tuning the masterpiece. After translation and folding, some proteins undergo further modifications called post-translational modifications. These modifications are like adding the finishing touches to a masterpiece, fine-tuning the protein's activity, stability, or localization within the cell. There are many types of post-translational modifications, including phosphorylation, glycosylation, and ubiquitination. These modifications can activate or deactivate the protein, target it to specific locations within the cell, or even mark it for degradation. Think of it like adding decorations, paint, or varnish to a finished sculpture, enhancing its appearance and function. Section 7. Protein synthesis in action. Examples in the body. Protein synthesis is a fundamental process that occurs in every living cell. Let's look at some examples of how protein synthesis is essential for our bodies. 1. Insulin production. Insulin is a hormone produced by the pancreas that regulates blood sugar levels. The insulin gene is transcribed into mRNA, which is then translated into the insulin protein. Defects in insulin production or signaling can lead to diabetes. 2. Muscle contraction. Muscle cells are packed with contractile proteins like actin and myosin. These proteins are synthesized through protein synthesis and are essential for muscle movement. 3. Immune response. Antibodies are proteins produced by immune cells that help fight off infections. The production of antibodies is a complex process that involves protein synthesis and is crucial for our immune system to function properly. These examples highlight the diverse roles that protein synthesis plays in maintaining our health and well-being. Section 8. Errors in protein synthesis when things go wrong. While protein synthesis is a remarkably precise process, errors can occur. These errors can lead to the production of non-functional proteins or even harmful proteins that can cause diseases. Mutations in the DNA sequence can lead to errors in transcription or translation, resulting in the production of faulty proteins. Environmental factors like radiation or exposure to certain chemicals can also damage DNA and disrupt protein synthesis. Some diseases are directly linked to errors in protein synthesis. For example, cystic fibrosis is caused by a mutation in the gene that codes for the CFTR protein, which regulates the transport of salt and water across cell membranes. This mutation leads to the production of a misfolded CFTR protein, resulting in the symptoms of cystic fibrosis. Section 9. Conclusion. A symphony of cellular processes. Protein synthesis is a fundamental process that is essential for life. It is a complex and elegant dance of molecules involving the coordinated action of DNA, RNA, and ribosomes to create the proteins that carry out the myriad functions of the cell, from enzymes that catalyze chemical reactions to structural proteins that provide shape and support. Proteins are the workhorses of the cell. Understanding the intricacies of protein synthesis is crucial for unraveling the complexities of life and developing new strategies for treating diseases caused by protein dysfunction. Like a symphony orchestra, each component of protein synthesis plays a vital role in creating the final masterpiece, a functional protein.